Back students, today we are going to study about spermatogenesis. So before starting spermatogenesis, we will do a quick revision of gametogenesis, which we have already discussed in our previous videos, previous classes. Okay, so let's start. Gametogenesis. Gametogenesis means formation of gametes. Okay, formation of gametes. Okay, and the gametes are of two types. Gametes are of two types. First one is male gamete, and the second one is female gamete. So male gamete is sperm, which are produced by a process of spermatogenesis, and female gamete is ova, which is produced by a process of oogenesis. Okay, there are two types of gametes, male gamete and the female gamete. Male gamete are produced by a process spermatogenesis, and the female gamete is produced by a process of oogenesis. In oogenesis, ova is produced, and in spermatogenesis, sperm is produced. A mature spermatozoa is produced. Okay, so we are going to study about spermatogenesis. We are going to study about spermatogenesis. So, what is spermatogenesis? It is a process. It is a process by which haploid spermatozoa developed from the germ cell from germ cells in the seminiferous tubule. In the seminiferous tubule. And where this seminiferous tubule is present? This seminiferous tubule is present in the testis. So, in the seminiferous tubule of the testis. Okay. Spermatogenesis takes place in the seminiferous tubule. Spermatogenesis takes place in the seminiferous tubule. Seminiferous tubule are lined by a germinal epithelium. As you able to see the structure here, when we take a section of testis, we will find a seminiferous tubule. On the inner side of the seminiferous tubule, there are the two types of cell. First one is germ cell, and the second one is conical cell, which is known as Sertoli cells. Okay, this one is germ cells, and the second one is Sertoli cells, which is conical in structure. Okay, these germinal epithelium were present on the inside area of the seminiferous tubule. Okay, so now let's talk about the stages of spermatogenesis. Stages of spermatogenesis. Okay, there are the two stages of spermatogenesis. First one is First one is formation of spermatids. In first stage, formation of spermatids takes place. So first one is formation of spermatids, and the second one is spermiogenesis. What is spermiogenesis? Here one question arises. What is spermiogenesis? So beta, it's a process. It process where spermatids differentiate into mature spermatozoa, or you can call sperm. Okay, spermiogenesis is a process where spermatids differentiate into mature spermatozoa. Okay, and formation of spermatids. In this stage, what happens? What are the functions, okay, of this stage? So beta, in the formation of spermatids, spermatogonia divide to produce spermatids. Here spermatogonia divide divides to produce spermatids. Okay, after the formation of spermatids, spermiogenesis takes place where the spermatids differentiate into mature spermatozoa or sperm. Okay, so now better let's see how how the process of spermatogenesis takes place, how these sperms are produced. Now just have a look. Okay, the spermatogonia, which is known as the sperm cell. Spermatogonia. This one is the germ cell. Okay, undergoes mitosis. Undergoes mitosis to produce two type of cells, type A and type B. Type A is spermatogonia. This spermatogonia produce type A, that is the spermatogonia only. Type A will produce spermatogonia, more spermatogonia, and these act as a stem cell. Okay, so it will produce more spermatogonia. Okay, type A will produce more spermatogonia and type A is spermatogonia. Spermatogonia. And this will produce more spermatogonia, act as a stem cell. Okay, act as a stem cell. And type B is primary spermatocyte. Okay, type B is primary spermatocyte, and here the chromosome number will be same. Suppose this one is the diploid. Spermatogonia, the chromosome number is diploid, and here also a diploid, and the primary spermatocyte is also a diploid in nature, diploid chromosomes. Then the chromosomes are diploids. In mitosis, the chromosome numbers remain unchanged. Okay, the chromosome number never changed. Okay, in mitosis. Here the primary spermatocytes are formed, which is diploid in nature, and this will undergoes meiosis division, meiosis 1. Meiosis 1. Okay, and this will produce two secondary spermatocytes. Two secondary spermatocytes. Okay, two secondary spermatocytes were produced after the division of meiosis 1, and as we all know that meiosis 1 is a reductional reductional division. In this, the chromosome number changed. The primary spermatocytes are diploid, and here the secondary spermatocytes are haploid. The chromosome numbers become half. Okay, this after a secondary spermatocyte, these secondary spermatocytes undergoes meiosis 2, and We all know that meiosis II is equivalent to mitosis. Meiosis II is an equational division where the chromosome number doesn't change. Okay, and here four spermatids after meiosis II, four spermatids are produced. Produced. Four spermatids are produced, which is haploid. Okay, and these spermatids are non-motile cells. They do not look like sperm. So there is a second stage of spermatogenesis. There is a process of changing spermatids into a mature spermatozoa, which is known as spermiogenesis. Spermiogenesis. Okay, spermiogenesis. After the process of spermiogenesis means the formation of sperms from spermatids, or you can say that spermiogenesis is a process where the spermatids differentiate into mature spermatozoa. Here sperms are produced. Sperms are produced, or you can say spermatozoa. Okay, let's see the structure. Suppose this one is the seminiferous tubule, and there are the two types of cells. These are epithelium, germinal epithelium, where the two types of cells were present, germ cells and the conical cells. Okay, which we have already discussed. As we are able to see here, Sertoli, this one is the Sertoli cells, and this one is germ cell. Okay, now say this. These germ cell divide, undergoes mitosis division and produce spermatogonia. Produce spermatogonia. Suppose this one is spermatogonia. Spermatogonia. Okay, the spermatogonia undergoes mitosis division to produce two type of cells, type A and type B. Okay, type B is beta primary spermatocyte, spermatocyte, primary spermatocytes were produced. Okay, which is diploid, because mitosis division takes place. Here the spermatogonia is diploid and the primary spermatocytes are also diploid. These primary spermatocytes undergoes meiosis division to form two secondary spermatocytes. These are the secondary spermatocytes. spermatocytes. Okay, secondary spermatocyte, which is haploid. Meiosis and meiosis 1. Okay, this will undergoes meiosis 2 division to produce 4 spermatids. These are spermatids, which are in haploid condition. Okay, this doesn't look like a sperm. This spermatid doesn't look like a sperm. These are non-motile. That's why these there is a need of changing the spermatids into motile condition. So there is a process which is known as spermiogenesis. Okay, to produce mature spermatozoa, and this will produce mature spermato s- mature spermatozoa. After the formation of these spermatozoa, this primary spermatozoa, these spermatozoa travels to here Sertoli cells, and these spermatozoa embed their head in the Sertoli cells. Okay, these spermatozoa embed their head inside the Sertoli cells. Why? To get the nourishment, to get its nourishment, because Sertoli cells provide nourishment to the spermatozoa. Okay, Sertoli cells provide nourishment to the spermatozoa. That's why these spermatozoa embed their head inside the Sertoli cells to get its nourishment. Okay, so let's write. Last point. After the formation of sperm, these sperms gets its sorry gets its nourishment from the Sertoli cells by embedding their head inside the Sertoli cells. I hope I am able to make you understand the things. Okay.